Just want to welcome everybody from the School of Medical Professions, the High School for Medical Professions. And I just wanted to say to you that you are now in our brand new School of Public Health building. And it is a phenomenal building, great study, great research, great lecture space. And it's space that we use not only for the institution, but for different types of events such as this. So I intend to invite you all for different types of programs, exposure to health career programs in the future, and we will be utilizing this building. As you see, it's a beautiful space. Don't you agree? Yeah. All right. So enjoy the program. Thank you.
Good morning, everyone, and welcome to SUNY Downstate. My name is Kari. I'm one of the students here. And just want to let you know, we're the only academic medical center in Brooklyn. And Downstate has been around since the 1860s. It was here before the Brooklyn Bridge was even built. And today we have 5 schools. We have the School of Public Health, the College of Health-Related Professions, the College of Medicine, the College of Nursing, and the School of Graduate Studies. And we're very glad that you could all join us here today to celebrate Dr. Furchgott's discoveries. And some of Dr. Furchgott's family is here on campus to take part in the festivities, so you'll see them around. And we're very grateful that they could join us as well. And now let me tell you about our speaker. So Dr. Penington obtained his PhD in pharmacology from the University of Alberta, Canada, and after obtaining his PhD, he did his postgraduate work at University of Edinburgh, Scotland, and the University of Chicago. And as an associate professor of physiology and pharmacology, he studies regulation of serotonergic neurons and more generally the control of neuronal ion channel activity. And when he's not doing research, Dr. Penington teaches neuroscience, biochemistry, and pharmacology at SUNY Downstate. So everyone, please welcome Dr. Penington.
Thank you. Thanks very much, Katya. So what I will try to do in the next 30 or 40 minutes is give you sort of an overview of what Dr. Furchgott did to win the Nobel Prize. And you can see this is taken from the website of the Nobel Prize. You can, and you can see that he actually was awarded the Nobel Prize 1/3 with Dr. Luis Ignarro and Dr. Ferd Murad. So I'll talk a little bit about what they did as well to discover the role of nitric oxide in the vasculature. Now, I think it's probably best if we get a feel for who Dr. Furchgott was, a very modest man. Winning the Nobel Prize really didn't change him at all. And so we'll watch a short movie. It's about 3 minutes.
2.5 years ago, I had occasion to look up nitric oxide. Then, just by chance, I looked across at the opposite page, and there was Nobel, Nobelist, Nobel Prize. And practically, when the dictionary is closed, Nobel Prize and nitric oxide almost kissed one another. And I said, maybe it's an omen.
In my work, I simply have been interested in finding answers to things that pique my curiosity, that come up. You can't predict what's going to come up and what the questions are that you're going to try to answer, but they develop those questions as you do more research, and you then try to get an answer to those questions. You never get a complete answer. You get a partial answer, and then the next step is what is a more complete answer, and so on. You use chemical agents, drugs, and so on, in certain ways to try to solve puzzles about how tissues function. And this is somewhat old-fashioned compared with molecular biology now and all the genetic studies and so on. But it still is useful and in terms of developing agents that are going to be of practical use. That's not my aim, but it turns out that pure research begins to get the answers to problems that then are helpful in developing new drugs and new procedures in medicine. The new generation of pharmacologists, physiologists, biochemists who do so much in molecular biology are going to make further breakthroughs in areas that, with my training, I can't get into, but I know are very interesting. The New York Times called me and said they have a report here from Associated Press and that I am the winner of a Nobel Prize. You don't do research to win prizes. It happens that you do win prizes sometime if your research is considered really important. I've worked hard and long and thought hard and long and had fun, and it's almost like the icing on a cake. The cake is good, that's the fun part. Yes. Now there's some extra icing on it, and it makes it even that much sweeter.
On behalf of the Nobel Assembly, I wish to convey to you our congratulations, and I now ask you to step forward to receive your Nobel Prizes from the hands of His Majesty the King.
You now grasp of the significance of the context of what Dr. Furchgott did and what he discovered. And so to do that, I'm going to try to start with a little bit of background. Okay, so you've all heard of neurotransmitters. They're chemicals that are thought to be released. Well, we know they're released from nerve cells. And this is an artist's rendition of a nerve cell terminal. That is the swelling at the end of a nerve. And when the nerve is stimulated, it releases chemicals, which this artist has drawn as a sort of yellow blob. And that chemical diffuses across a gap and activates a muscle postsynaptically or a nerve. And the chemicals bind selectively to a receptor protein in the postsynaptic membrane. And it's sort of like a lock and a key situation, where the chemical is the key and the receptor protein is the lock. Once the lock is activated, the receptor is changed in some way, and that causes a biochemical reaction to occur postsynaptically in the cell. Okay, so neurotransmitters are very important because they're released by the nervous system, and you can think of the autonomic nervous system, which is the nervous system that exists outside of the brain, even though it's controlled by the brain. You can think of it as having two sides to it. A side that is activated when you're stressed or when you're fearful or you need to fight. And then there's a side which is activated when you're very calm. So the sympathetic side is the side that's activated when you're stressed, and you can think of it like an accelerator on a car. It accelerates everything. It causes, the neurotransmitter is norepinephrine, and it causes your pupils to expand, to allow more light in. It causes you to breathe more shallowly but more frequently. It causes your heart to beat much faster, and it decreases the activity in your gut. So you're ready for a stressful situation. But there's another side to the autonomic or peripheral nervous system that calms you down, and it acts a bit like a brake. And acetylcholine is thought to be the neurotransmitter. Well, we know it's the neurotransmitter. It causes your pupils to shrink and let less light in because you're really focusing on things that are up close. It causes your breathing to slow down and be deeper. It slows down your heart rate, and it activates your gut because when you're relaxed, you can digest your food. So this is really the key here, that Dr. Furchgott figured out how acetylcholine works pretty much at the molecular level. And so at least one aspect of how acetylcholine works, an important aspect, the way in which acetylcholine affects the blood vessels. Okay, So here we have acetylcholine that's contained in sort of lipid vesicles that fuse with the presynaptic membrane of the nerve. The acetylcholine transmitter diffuses across the synaptic gap and interacts with the receptor protein, the so-called muscarinic receptor, on the postsynaptic membrane. Now, there's one other piece of background that I'd like to go over before we delve into what Dr. Furchgott did, and that is the structure of blood vessels. So here we see on the left an artery and a vein. And you can see, well, you should be able to see right away that they're made up of layers. This layer here is a smooth muscle layer, and that can contract or it can relax. And what was known about acetylcholine in the past was that it causes this smooth muscle to relax so that the blood vessel opens up and allows more blood to go through it more rapidly. And that lowers the blood pressure. Okay? So in addition, and this is the same for veins, but in addition, you can see that there's a single cell layer on the inside of the artery called the endothelial layer. And at the time that Dr. Furchgott was doing his work, nobody really understood how the endothelium worked in blood vessels. And it was his major contribution to show us what the endothelium contributes towards the contraction and the relaxation of blood vessels. Okay, so as I pointed out, Dr. Furchgott knew that acetylcholine caused blood vessels to relax. And he knew this because when you inject it into the limb of an animal, the blood vessels relax and the blood pressure in the limb drops and the perfusion increases. But what he wanted to do was to create an isolated preparation of an artery that he could string up with a forced transducer so that when it contracts or relaxes, he could measure it. Okay, so what he did was he developed a helical strip of the rabbit aorta. The aorta is a large artery that it comes from the heart and sends blood around the body. So you can cut out a large piece of artery from an anesthetized animal, such as a rabbit, and he cut a helical strip. So he cut around the artery to make a helical strip of the artery, and then he was able to string it up in an apparatus which I'm going to show you now. It looks complicated, but actually it's pretty simple. So what it consists of is a water bath with water and a heater and a thermostat so that you can control the temperature of the water. And then we have an organ bath here, a smaller bath, with sort of like a fish hook at the bottom and a fish hook at the top that you can attach to the piece of artery. And then a piece of string is attached to a force transducer, which can measure the force that the artery will generate in response to a drug. So when it contracts, the transducer moves a pen, which is on a moving drum that has paper on it. And so you can measure the contraction or the relaxation of the artery when you inject the drug into the bath at a known concentration. So that's really it. You can change the solution that the artery is exposed to by perfusing it in this way. So not a very complicated but very useful preparation. Now, so Dr. Furchgott did this with some helical strip of artery, and he got a very strange result. When he added the acetylcholine, it caused contraction of the blood vessel. And that was very surprising to him. He couldn't explain it. So he tried to do the experiment every which way he could think of doing. He tried to relax the artery first, put acetylcholine on. It caused a contraction. Then he tried to stimulate the artery so that it was contracted. Then he put the acetylcholine on. It caused contraction. Now, this is not supposed to happen because, recall, when acetylcholine is injected into an animal, it causes vasodilatation, relaxation of the artery. So after a while, at this time in 1953, which was 3 years before he came here, he couldn't figure out what was going on. So he put the problem aside and he worked on the math of drug receptor interactions. So how drugs interact with receptors and produce their response. And he became a rock star for that work. He's quoted in all the textbooks. So I knew what he had done. When I came here in 1993, he actually interviewed me for my job, one of the people, and so I knew what he'd done, and I also knew about his work on nitric oxide because he'd published that in 1986. So that really helped, I think. It certainly didn't hurt. So he put that aside and then fade back in 25 years later. He put this aside for 25 years. He was interested in looking at the effects of the accelerator transmitter, norepinephrine. He knew that norepinephrine causes contraction of blood vessels, but when you block a certain type of norepinephrine receptor, you can also see another effect mediated by a different receptor for norepinephrine, a relaxation. And he wanted to study the relaxing effects of norepinephrine. So he instructed his technician to put up a preparation of this rabbit aorta and then put on norepinephrine, get a contraction, block it, and then put on acetylcholine to get a contraction so that he could then block these receptors he was interested in that block the contracting effects and look at the relaxing effects of norepinephrine. But the technician did the experiment in the wrong order. He put on the norepinephrine, he got a contraction, and then he put on acetylcholine, and this is the result that he got. So here is the norepinephrine. It caused contraction of his artery. Then he added acetylcholine in increasing concentrations, and he saw a relaxation. This blew Dr. Furchgott's mind because he'd never seen this before. And this is what you're supposed to see with acetylcholine, the relaxation of blood vessels. And if you'll recall, 25 years before, he had published that acetylcholine under all conditions causes a contraction of the artery. And he realized that this was very, very important. And it didn't really matter whether he disproves his previous work. He had to figure out why this was happening. And the reason, when he figured out why it happened, he won the Nobel Prize. So he actually now started to try to think very seriously about what was going on here. And it took him 2 weeks to figure out what was going on. He realized that instead of using a helical strip of the artery, they were cutting the artery across to make a ring. And they were just stringing up the ring, and when they put the acetylcholine on, it caused relaxation instead of contraction. So he got the people in his lab to put up helical strips, and they put on the acetylcholine, and it caused contraction. So they got the opposite effect depending on how they prepared the artery. And they realized quite soon that the way they were preparing the artery, they were doing damage to it. They were actually putting glass rods into the artery and rubbing away the endothelial cells, that thin one-cell layer. So here's the data. They compared the ring with the strip, and you can see when the inside of the artery wasn't rubbed, they got inhibition to acetylcholine, okay, irrespective of whether it was a ring or a strip. When they rubbed the inside of the ring, they either got nothing or a small contraction with acetylcholine. And then they added an enzyme which could strip away that single layer of cells inside an artery. And when they did that, adding collagenase, now they were able to get contraction in rings or in strips. So the conclusion that they made was the endothelium was playing a major role in whether or not you see a contraction or an inhibition to acetylcholine. Now I'll give you a little bit of history, which Dr. Furchgott was very well aware of. Back in the 1920s, Sir Henry Dale was figuring out what you need to do to prove that a chemical is a neurotransmitter. And he had in his lab in London a German fellow by the name of Otto Lowy. And as very often is the case in science, Otto Lowy had a dream about what experiment he should do to prove that a chemical is a neurotransmitter. And so what he did was he got an isolated frog heart with the vagus nerve attached to it. And when you stimulate the vagus nerve, the heart slows down its rate of beating. And he strung up the heart above a second heart that didn't have the vagus nerve attached to it, and then perfused the first heart so that the perfusate dripped from the first heart onto the second heart. When he stimulated the nerve, the vagus nerve, it released what he called was vagus stuff, which we now know is acetylcholine. And the perfusate then dropped onto the second heart and slowed the rate of beating of the second heart. So with that experiment, they proved that chemicals get released from a nerve and they can travel from one preparation to another and influence the activity of the second preparation. And that's called a bioassay. So Dr. Furchgott decided he would do a bioassay, and this is what he did. So he set up the preparation of the rabbit aorta, which has had the endothelium stripped away from it. So when you contract it with norepinephrine, then you apply the acetylcholine, you get a contraction. Okay? So then he took a second piece of rabbit aorta where he didn't strip off the endothelium, and he sandwiched it against the previous artery. So when you stimulate this tissue, it stimulates the endothelial cells, which released something onto the previous artery that would only contract. And this time, when you added acetylcholine, you got relaxation. So what this elegant experiment did was it proved that the endothelial cells here were releasing something that caused relaxation of the blood vessel. And he called it endothelial-derived relaxing factor. And this was published and was very widely known at the time. Okay, so about this time, no work happens in a vacuum. We all stand on the shoulders of somebody else in science. About this time, Farid Murad had shown that you could activate a chemical in smooth muscle which would result in relaxation. And this is called the Cyclic Gmp System. So this is a base attached to a sugar molecule with 3 phosphates. And this is acted upon by an enzyme, guanylyl cyclase, which cyclizes the phosphates to form cyclic GMP. So it turns out that when you activate the production of cyclic GMP in smooth muscle, you cause relaxation. And Dr. Farid Murad had actually shown that when you put acetylcholine on smooth muscle, it activates cyclic GMP, and this correlates with the smooth muscle relaxation. So this is data from Dr. Furchgott. He went ahead and did the same experiment. So here he shows the relaxation of smooth muscle caused by acetylcholine. And he measured, at the same time, the production of the cyclic GMP chemical. And you can see it occurs before and has a similar time course as the relaxation of the smooth muscle. Okay? Let me just see. Okay. About the same time, Farid Murad was also interested in how glycerol trinitrate, or nitroglycerin, works to relax blood vessels in the heart. And he thought that nitroglycerin was likely to release nitric oxide within the tissue and to activate cyclic, the enzyme that generates cyclic GMP, which then leads to smooth muscle relaxation. So in fact, Farid Murad went ahead and proved this sequence of events. So then the major question that occurred to Dr. Furchgott was, does acetylcholine also stimulate the endothelial cells to produce endothelial-derived relaxing factor, and could it be nitric oxide, which then penetrates through the cells to activate the muscle cells and cause relaxation? Okay. So about this time, some work was being done in Glasgow in Scotland on nerves which resulted, activation of which caused relaxation of smooth muscle, and they called it non-adrenergic, non-cholinergic nerves. They couldn't figure out what the neurotransmitter was, so they called them NANC nerves. And I remember being taught about this when I was studying pharmacology. And a postdoc, William Martin, came over to Brooklyn from Glasgow and worked with Dr. Furchgott to show that when you add norepinephrine and you contract the artery, then relax it with acetylcholine, if you add hemoglobin, which is the protein in the blood that carries oxygen, you can completely reverse the relaxing effect of acetylcholine. And so he thought that the hemoglobin was somehow interacting with endothelial-derived relaxing factor and mopping it up so that it couldn't activate the guanylyl cyclase and produce cyclic GMP to cause the smooth muscle relaxation. So that work with William Martin showed that the acetylcholine effects on the smooth muscle were all blocked by hemoglobin. Right. And that also the hemoglobin blocks the effect of cyclic GMP as well in the smooth muscle. So just a few words about free radicals. I'm not sure how much chemistry you've had, but I think to understand what's going on here, I need to show you the molecule oxygen. So here we have 2 atoms of oxygen, and each atom has 6 electrons of its own. And that's not a very stable situation because oxygen, to be really stable, needs 8 electrons in its outer shell. So what the molecule of oxygen, O2, does is it shares 2 of its electrons with its neighbor. Okay? So what this means is that oxygen really wants to accept 4 electrons. And that's really the basis of life on Earth, because oxygen ultimately accepts electrons throughout biochemical reactions. And that's why we need oxygen to live. So, as I said, oxygen wants to accept electrons. So if you add an electron to oxygen, you get O2-, which is called a superoxide radical. And it's very reactive, and it wants, to donate that electron to another atom somewhere else and cause chemical reactions. Okay, so that's all we need to know about free radicals. Well, the next piece of the puzzle was contributed by Salvador Moncada, who actually did a lot of work with nitric oxide, but he didn't win the Nobel Prize. So what he showed was that superoxide free radicals can interact with nitric oxide and quench it to form peroxynitrate. So in other words, if there's any free radicals of oxygen around, it will prevent the effect of nitric oxide. And there's an enzyme that we all have that actually mops up these superoxide free radicals, converts them to hydrogen peroxide and water. So this natural enzyme will prevent the quenching of nitric oxide. And Dr. Furchgott knew about this work, and he used it in his studies. So Dr. Furchgott also had a prepared mind because he knew his inorganic chemistry. He knew that if you take sodium nitrite and you acidify it, you get HNO2. The HNO2 can break down to produce nitric oxide. So he realized that you can add sodium nitrate to a muscle as long as it's acidified, and you can generate nitric oxide within the bath. And that's the experiment that was done by Dr. Jyothyanandan, who's in the audience here. Would you like to stand up and say hi? Okay, so here's the experiment that Dr. Jyothyanandan did. So here we have the control where the sodium nitrate, the acidified sodium nitrate, has exactly the same effect as acetylcholine. It causes relaxation of the smooth muscle. If you add the enzyme superoxide dismutase, which remember destroys all the superoxide free radicals, it potentiates the effect of nitric oxide because it can't bind to the nitric oxide. So now we see that the response is much larger. You can do the opposite effect by promoting the generation of these superoxide free radicals. And when you do that, you see that the response is drastically reduced. So there was one more experiment that needed to be done to prove to Dr. Furchgott that endothelial-derived relaxing factor was in fact nitric oxide. And in fact, although the stage had been very well set, nobody had actually suggested that endothelial-derived relaxing factor was nitric oxide until really the next experiment was done. And this was also done by Dr. Jyothyanandan in Dr. Furchgott's lab. So what they did was they took the bath solution, the salt solution that the muscle sits in, and they bubbled it with nitric oxide so that it was saturated with nitric oxide. And then they took a very small amount of the nitric oxide, 2 microliters, and they injected it into the bath that contained the smooth muscle artery. And you can see that when they injected the bath solution that was bubbled with nitric oxide, they got responses that were dose-related and that looked exactly like that of acetylcholine. And then when they added the enzyme superoxide dismutase, the same concentration gives a much bigger response. Okay? So this was enough to prove to Dr. Furchgott that endothelial-derived relaxing factor was in fact a gas which is itself a free radical called nitric oxide. And he published this at a meeting and told the world that EDRF was nitric oxide. But then about 2 papers later, at the same meeting, Dr. Luis Ignaro made the same claim. And he did it from a different angle altogether. And this was a very clever experiment. What he did was he looked at hemoglobin, and he looked at its ability to absorb light. And you can see here with the orange deflection, this is the hemoglobin absorbing light. He did it in the presence of tissue that was exposed to acetylcholine so that EDRF, the endothelial-derived relaxing factor, was produced. And you can see that it shifted the absorbance of the hemoglobin to this deflection that's in green. So then he did the experiment where instead of exposing the hemoglobin to EDRF, he exposed it to nitric oxide. And he got exactly the same result. So the deflection caused by hemoglobin was shifted by nitric oxide in exactly the same way that EDRF shifted the absorbance of hemoglobin. So he said this is evidence that EDRF is nitric oxide. And both of them published their results about a year later, and that would be, I think, 1987. So again, Salvador Moncada finished off the theory here. So acetylcholine interacts with the receptor on the smooth muscle. It causes an increase in intracellular calcium, which then activated an enzyme that was discovered, I think, by Salvador Moncada, called endothelial-derived endothelial nitric oxide synthase. Now, this enzyme converts an amino acid that has an amine group. It releases nitric oxide from that amino acid. The nitric oxide then diffuses into the smooth muscle cell. It activates the cyclic guanylate cyclase to activate cyclic GMP, and that's what causes the relaxation of the smooth muscle. So all that remains now is for me to say a few words about how important this discovery was. So this allowed an understanding of how nitroglycerin works in coronary artery disease. So in coronary artery disease that causes angina, which is a tremendous pain that comes from your heart, it's because the blood vessels are too constricted. And so by giving nitroglycerin, it generates nitric oxide within the smooth muscle of the blood vessel, and that causes the stimulation of cyclic GMP and relaxation of the blood vessel so that the heart can now be perfused with more blood, and it relieves the pain. Nitric oxide is also important for signaling between nerve cells in the brain, and it's also been discovered that nitric oxide is involved in our normal defense against bacterial and parasitic infections. So nitric oxide can be generated within, in the vicinity of bacteria, and the nitric oxide is a free radical and it damages the bacteria and contributes to the destruction of an infection. And finally, you can just see the very direct effect of nitric oxide. Here we have a newborn who has very high blood pressure because the blood vessels are constricted in this infant's lungs. So if the infant is allowed to breathe nitric oxide, it causes vasodilation of those blood vessels and allows the baby to breathe and survive. So clearly, there are many, many applications of this discovery, and I think that this is really obviously the main reason why this discovery merited a Nobel Prize. And finally, Alfred Nobel, it's kind of ironic, he's the guy who discovered a nitroglycerin, which is an explosive liquid. And so he invented this explosive and made millions and millions of dollars, which he then invested and created the Nobel Prize for Science with. Later in life, he got angina pectoris, which is this pain in his heart because he had blocked arteries. And they prescribed nitroglycerin to relieve his angina pain. And he wrote to a friend, "It sounds like an irony of fate that I have been prescribed nitroglycerin internally. They have named it trinitin in order not to upset pharmacists and the public. Your affectionate friend, A. Nobel." Okay, so that's really all the material I have to explain to you about why this discovery by Dr. Robert Furchgott resulted in the Nobel Prize. Thank you. Any questions? That's a very good question. All I can assume is that there's more than... Okay, so Dr. Wong, the current chairman of the Department of Pharmacology, Dr. Furchgott was the chairman of the Department of Pharmacology at the time that this work was done. So Dr. Wong asked me why acetylcholine causes a contraction when the endothelial cells are removed. Because clearly there is probably more than one muscarinic receptor in the endothelial cells and in the smooth muscle, actually in the smooth muscle. And we know that certain muscarinic receptors result in the stimulation of calcium within the cell, which then activates the NOS enzyme to generate nitric oxide. But other muscarinic receptors actually don't raise calcium, they lower calcium. So it's entirely possible that in the absence of the nitric oxide that's produced by the endothelium, that this smaller response wins out, and you see contraction when you remove the endothelium and you don't see the effect of nitric oxide, obliterating it. But clearly, the physiological important response is the response that you get when the endothelial cells are still present. Any more questions?
Any questions from our students?
And I might not be able to answer them all because actually this is not my field. Still. Well, if that's the case, I'll let everybody move on. Thank you.
Interesting enough, though, I thought it was very, I was listening to the lecture, I'm like, wait a second, can I get it? When you hear something for the first time, it may not click. But you hear it the second time or another time, it's all going to come together. So I really, really hope that you're able, students, to take some things away with you today. And I do sincerely want to thank Dr. Penington for taking the time today to come out and give us this talk. So let's give him another round of applause. I just wanted to make a couple announcements. The students, what's going to happen is that we're going to have you tour the gallery in full. That tour, you're going to see a lot of different things, and there'll be other employees around that may be able to answer your questions if you have questions about that. We are giving you small booklets. You'll get a chance to see In those booklets are descriptions of those various awards. And later on, when you're back home, you can actually take the time to go online and look and read about that particular award. What happens is that when you get an award, people get to know about you, and they say, wow, you know what? I really want to be part of that person. So you get more awards and more awards and more awards. And over time, Dr. Furchgott received a number of awards for his basic science research. Basic science means that we're looking at things from a simple point of view so that we can look at it a larger point of view. So you're going to get a chance to see a lot of medals and certificates, and there's some statues and things like that. And if you don't get it all today, you can certainly read about it in the book, and you also can go online. Following that, you're going to follow directions from Michael Harrell, who's going to have you take a picture with our president. Okay, I'd like the Furchgott daughters to please come up. Please come up, everybody. Please give them a round of applause. Sorry it didn't happen. That's my fault. All right. Yeah. We're lucky that the Furchgott daughters flew in from all parts of the country to be with us today. They're also going to be in the gallery, so you could ask them all kinds of personal questions about their father. All right, and we get a chance to hear about their father that way. So, but we do have a number of faculty members and staff members who will be happy to answer your questions. This room is secure. You can leave your things in this room, and you can go to the gallery. Michael will instruct you. So this room is secure. I know you're taking your phones with you. You should, so that you can take pictures. Okay. Did you have anything else, Michael? Okay. Well, thank you, guys. We'll see you out there. Oh, and I'm sorry. Go ahead.
Yeah, well, it's really nice to
Come
see
over here.
students here interested in science, because I was the oldest child of Dr. Furchgott, and he really wanted me to be a scientist. And I got good grades, but all of us became artists. And you're the people that are following the sciences. And so Brooklyn is a great place. I'm so glad that he got the award, because Downstate, I even worked here sometimes in the summer. It's just so important, all the work that you're doing and that you're going to do in the future, because it all helps us. It all helps people. And, I didn't understand everything in this lecture, and maybe you did, but you're going to go way ahead of any of us. So thanks for being here.
All right. Okay, guys.